I work with fish and mussels across the state of Mississippi. Usually that involves listed species, either federally or state listed species, but we work with basically all the species across the state, so we get to see a lot of really neat stuff. As curators here at the museum, we kind of have a bunch of different hats we wear. Not only do we sit in the back and browse over dusty, old, nasty collections with tags that you can barely read, but we also go out and collect our own and add to the collection. So it's a growing collection of knowledge for the entire state of Mississippi. So, if you don't know, the Mississippi Museum of Natural Science was established in 1939 by Fanny Cook. She was actually a school teacher in Mississippi and saw a vast lack of knowledge in the state's biodiversity. We didn't have any sort of state fish and game commission, so there were no regulations on any take or harvest of our species across the state, and we had no kind of repertoire of knowledge on what we even had in the state. So she started collecting across the state. She actually funded her own conservation commission that went across the state and started collecting with the WPA in the early 1930s. A lot of species of fish, birds, mammals, they kind of just took everything they saw. And then in 1935, she established helped establish by state legislature the Fish and Game Commission for Mississippi, and then established the museum in 1939, where she led as the director. So our museum houses both a public exhibit area as well as several biological collections, which includes fish and mussels, which I help curate. And Fanny Cook was actually our first ichthyologist and wrote The Fishes of Mississippi right here in 1951. So how do we start these collections? So either back in the day or nowadays, the first thing you got to do is go out and collect something. So you go out with various different methods depending on the taxa or depending on the species, and you go out and collect it. Then you got to preserve it. Thanks, Matt Wagner, for this animation. Throw a tag in there. So with fish and some of our mussels, we preserve them in fluid, and the way you do that is you get a live fish, you humanely euthanize it, you put it in formaldehyde, that soaks in there a couple weeks, that fixes the fish, you soak it in water, and then you put it into ethanol where it lives on forever. And with that, you have a tag that goes in that has all the information that you could want for that little collection. And that gives us cool things, like we can go back and look at genetics on some of these things, we can look at diet, you can look at parasites, taxonomy, distribution, all the way back. I think we've got specimens going back to the 1800s, so you can look at this through time, throughout the state, which is a really cool and powerful tool to have. In the collections back in the back, we have 100,000 lots of fish. Our oldest collection, like I said, is from 1888. And we have a fluid collection, a cold storage collection, tissue collection, and a bone collection. So a lot of different stuff back there. If you've ever been back there, had a chance to go back there, it's really cool. And this year we actually put in the 100,000th lot, which was the state record blue catfish. I think it was 141 pounds. And again, you have all that information for that. This individual we also aged. It was 25 years old, so you get a lot of information from just one specimen sitting back there. Same thing with mussels, 19,000 lots, oldest collection back to 1936. And we have our dry shell collection, which are what these guys are from up here, as well as tissue, cold storage, and stuff like that. So what do we do with those? All of that information goes into our database. We help manage a statewide database of both fish and mussels. With fish, we have over 240,000 collection records from across the state. And this database is not just from our museum, but we also brought in a lot of other museums and collections, including 31 university, state, and federal institutions, which includes things like the Smithsonian. And then same thing with mussels. We have 18,000 lots of mussels in our database from 43 different universities. And this is important because if you don't know, Mississippi and the Southeast as a whole is the Amazon of North America as far as freshwater diversity is concerned. We have some of the richest and most diverse freshwater fauna in the U.S. and in the world. And over 90% of our mussel species within the Southeast are endemic to the U.S. More than a quarter of its aquatic species are found nowhere else in the world. And then we also have a high rate of impairment because over time we really haven't prioritized conservation. It's really only been in the last 50 or 60 years. We've really prioritized aquatic conservation and prioritized kind of preserving some of this diversity and some of this habitat across the Southeast. So in Mississippi, we have 287 species of freshwater fish. We also have 77 species that are currently thought to be in need of conservation. They're called SGCN species, species of greatest conservation need. And these are listed on our upcoming State Wildlife Action Plan, which kind of details what we plan to do with those species, how we plan to conserve them, what their needs are within the state. And these can be just species, and I'll talk about that in a minute, that, it's as much as maybe it doesn't need conservation, we just haven't seen it in a while, so we got to go look for it. Same thing with mussels. We have 89 species now in the state. I think before Matt Wagner and I started looking at mussels, we had 84, so we're coming up. We're getting more. But we have 45 of those, so almost half of those, or over half of those, are listed as SGCNs in the state. So mussels aren't doing that great in Mississippi. So where are they now? This is the important question, and this is kind of what my research revolves around in Mississippi. So we're updating distributions for all of these state-listed species, federally listed species, and just all the species in the state for both fish and mussels. The most important thing right now is most of our species have very limited data. Maybe they don't need conservation, but we just haven't looked for them in a while, so we have no idea if they do or they don't. We have species of conservation concern, so maybe they're not doing great, but they're doing okay. And we have state-listed species, so these are species are a little bit more conservation concern. We know they kind of need conservation, whether that just be status surveys or actual propagation, releases, stuff like that. And then we have federally petitioned species and federally listed species. And those species are kind of the most important, as we already have kind of knowledge that that species actually needs active conservation. So first, when we're starting our surveys, what do? We have to identify our target species. In 2021, we started a survey on the salt marsh top minnow. We determined our reason for sampling because this species was up for federal petition, meaning somebody petitioned the Fish and Wildlife Service to list this species under the Endangered Species Act. So our reason for sampling was we believed that it was in decline across its range and within Mississippi, and we just didn't have a lot of data on it, so we needed to go look for it. We determined its historical occurrence, so where it used to be found across the coast of Mississippi. We looked at habitat preferences, so where this species prefers to be. What habitat is it utilizing, what time of year can you find it there, that sort of thing. And then we established a sampling protocol. We looked at gear, data collection, sampling time, what number of sites we wanted, stuff like that. And we actually determined that historical sampling was kind of inadequate. Historically, you kind of looked for these guys with a seine, with traps. Unfortunately, if anyone's ever seined in a salt marsh, you're in knee-deep mud, there's a lot of shells, a lot of bad material to get your seine hung up on, and it's really hard to chase a minnow that can see you coming because they live on the surface of the water. So we just thought that might be inadequate. We had heard Louisiana had some success with a shock boat. Usually, shocking occurs in freshwater. People boat electrofish for fish all the time. That's how we manage most of our freshwater systems. Not a lot of it's been done in saltwater. So we rigged up a boat electrofishing system, and we took it out and just decided to see what happened. We went and found it. If you can see, very hard to see, our number of efforts compared to historic sampling efforts. So basically an effort is how many times you pull a seine, or how many times, how many traps you set, stuff like that. For our 50 efforts, we caught 3,000 salt marsh top minnow. Some of these historic sites or collections, they had 2,000 efforts and caught 600. So we blew all these surveys out of the water with our new method of sampling for this species. And these species, their average number of individuals per collection, or these studies, like this one was 2 individuals per collection. Ours were 63 per collection. So you can kind of see these really skewed, that data going into this federal petition. It looked like that species isn't really abundant. Didn't really know it was that hard to catch, but you just kind of assume, oh, it's not very abundant. There's not a lot of them. They're not very widespread because we didn't get them at a lot of sites. It needs to be federally listed. But based on our study, I think it was the most abundant species that we caught. And because of our data, because of Louisiana's data, because we went out and looked for it, we kind of sampled for it the correct way, this species wasn't actually warranted for federal listing and didn't get listed, which saves a lot of money, time, and effort because you're not trying to manage this species when it actually doesn't need to be managed. So there's some pretty sites. Here's all the sites we did. You can see the green is where we got them, the red's where we didn't get them. So they're still fairly widespread across the coast. They are still listed on our state wildlife action plan because they are losing habitat. So even though they are very abundant, where there is habitat, there's not a lot of habitat left. You can see where they aren't right here. We didn't even sample because if you've been to the Mississippi coast, it's kind of just a sandy beach. Obviously, they're not going to be there. So they really don't have a lot of habitat across the coast, but where there is habitat, they're very abundant. And here's some photos from the field. There's us shocking. So what do we do with all that information? Well, we can publish it in gray literature. That includes our state wildlife action plan, some of our technical reports, some reports we just send out to the public, stuff like that. We make posters every now and then. We can do poster presentations where we're kind of at a scientific conference. We're presenting scientific data, but it's still not peer reviewed. It's still a little more unpolished data, just throw it out there. Oral presentations kind of like this, or at scientific conferences where you're presenting your data to the public for public comment. We can present peer-reviewed notes, which is still not a lot of effort. You're just doing a little bit of information you're putting out there, but it is peer-reviewed. Or you can do a full-on article, which takes a lot of time. We really don't have time for that, but some of our data actually does go on to be published in scientific journals, which is a lot of fun. So some of our recent projects, and this is kind of the bulk of my talk, just talking about all the cool stuff we've seen. I'd really like to get into the nitty-gritty of fish and mussels, and maybe that'll be another talk. But right now, we'll just kind of go on, some of these status surveys we get to do across the state and kind of appreciate some of the diversity that we get to see across the state. So in 2021, we were kind of messing around in the Big Black River. We sampled 89 sites. We found 3,000 live mussels, and that included 29 species of mussel, and 2 of those were federally listed species. One of them, this guy right here, the round hickory nut, was federally petitioned at the time, but it ended up becoming federally threatened afterwards, and our data actually went into that species status assessment. And then this is the rabbit's foot mussel, and that mussel is already federally threatened. So that was a lot of fun. If you've never been to the Big Black, it's really cool. It's one of the most interesting systems in Mississippi. You can see right here, these are all geological formations, and it goes through, I think, 13 different major geological formations as it gets down into the Mississippi River. It starts way up here in kind of north central Mississippi and works its way all the way down to the Mississippi River below Vicksburg. And it's really cool from a mussel standpoint because of the varied geology. You get all these different species in different places and different pockets depending on what geological layer you're in. So what the surface geology is, what the formation's kind of providing for those mussels. You get to see a lot of really cool stuff. So basically, this map shows the bigger the circle, the more species we found at that site. So there's not a lot through here because both of these geologic formations kind of just provide unstable sandy habitat, not a lot of mussel habitat. But up here you get a lot of clay and silt formation, and it's really slow and stable area, so you have a lot of cool different species of mussel, and that's where one of the federally listed species of mussel, the round hickory nut, is. And then down here you get into the Lurch Formation, which is a lot of gravel deposits that are coming into the river, so you get a lot of stable gravel shoals, and that's where we found that rabbit's foot mussel down here. And then as you move further on down, you get into the alluvial plain, kind of like the Mississippi Delta, so you get a lot of slow, deep silt areas, and you get a lot of cool mussels down here too. So it's really cool to go through, in 3-4 month span and just see all these different geologic formations, because you just get, it's a completely different river depending on where you're at. And here's kind of what it looks like doing that. That's about 10 or 12 feet deep. You're diving off of, and there are a lot of alligators in the Big Black. Fish and Wildlife Service biologists in this room can attest to how scary some of the gators are when they come up on you and you think that they're a real alligator. Here's some pictures from that. It's a lot of fun, a lot of mud, a lot of dirt, but we had fun. There's a boat ramp we had to dig out before we got our boat out of the water. And then it's just a really pretty river system. This is one of the prettiest areas in Mississippi. It's wild. There's no houses. There's no people. You're just out there on your own, so it's a lot of fun. In 2022, we went down to the coast again and surveyed for blue nose shiner. This is a really cool species that live exclusively in blackwater creeks. They're very pretty. They're very hard to get. You can see the habitat right here. Very deep, very slow. Here's Calvin and I kind of checking out some blue nose. There's more of their habitat. Really hard to sample for. Pearl River, we did the entire Pearl River in 2022 for mussels. It was a lot of fun. Yes, we dove below Jackson. Only one of us got a flesh-eating bacterial disease. So I think those are good odds. But this was a lot of fun. We ended up finding, we surveyed 118 sites across the river. We found 33 native species and 13,000 live mussels. So there's still a lot of mussels in the Pearl. We found 3 species of conservation concern. All of these 3 species are now federal conservation concern, including the Alabama hickory nut. We found 14 individuals. Unfortunately, they used to be right here in Jackson. They're no longer here, but they still are in the Occonookee River above Ross Barnett. So that was fun to see. And then the delicate spike. Unfortunately, this species hadn't been found in the Pearl River since 1978 alive, and we didn't find it alive. We found 2 fresh dead specimens in the Strong, but we're still hoping to kind of find some live ones holding out somewhere in the Pearl River. And then we also identified a new species to the state. So we found the Louisiana pigtoe, and we found it below Ross Barnett spillway. And that was a really cool species because we found it, and then it was also on the docket for federal listing, so our data went straight into that federal SSA as being in Mississippi, and it is right now proposed threatened, so it should be federally listed in the next year or so. And we actually got to go back within the collection and kind of prove that it's not just, a wave occurrence or a species that just happened to drop off a fish and, end up in the Pearl in the last decade or so. We went back and some collections that were identified as another species going back to 1936, we actually re-identified as Louisiana pigtoe. So it's been here. I think this was the 1936 collection right here at the museum, right at the Lowhead Dam. So it's been here since at least 1936. So this population is established, and it's also, it's been here for a long time. So that was really cool to see and use the collection in that way. Here's some field pictures. This is down in the lower Pearl River. You can see this massive gravel bar, and then the Pearl River is all the way over here. A lot of erosion down there, but it's not great for mussels, but it's it's very pretty area. Still muddy. We got a lot of mussels, a lot of dirt. There's Calvin sinking in the mud. I think there's a big alligator back here. Here's the Strong River. So got to see a lot of pretty stuff across the Pearl. This is the swinging bridge at Byram. And I think this is down below Columbia. So, Tennessee, last year we went up to one of the prettiest areas in the state, which was Bear Creek. It's a lot of fun. We found, of our 77 sites that we surveyed, we got to use a lot of cool different techniques. We kind of threw the kitchen sink. It was more of a status survey for the entire Tennessee River drainage than a specific species status survey. So normally we just look for a specific species or two, but this was kind of a, hey, let's just see what's here. So we got to do some kick seining, we did some backpack electrofishing, boat electrofishing, and then a lot of gill netting. So we got to see a lot of cool different species. We found 10,000 different fish, which included 96 species that we documented from those systems. And we found 24 of the 33 species of greatest conservation need that are kind of up in that area. So that was really cool. We had a lot of success doing that. Here's some really pretty, this is a gilt darter, and this is a northern studfish. We also found 2 species that were, where we didn't think we were going to find. We found a new species for the state. This is the purple lilliput. Calvin and I think it was our first site of the survey, walked down, and Calvin picked up a little mussel and was like, oh, hey, we found a mussel. And I looked at it and said, oh, that's a new species for the state. So that was really fun. And then we kicked up this little dude. We were in Cripple Deer Creek. If you don't know what a hellbender is, it's it's North America's largest species of salamander. They get about yay big. We have one little population known in Bear Creek, but we were in a system that's just above Bear Creek called Cripple Deer Creek. Didn't think we'd find hellbenders there. We ended up kicking up this little leaf pack, and we found a juvenile hellbender. So that was the first sign that hellbenders are recruiting or having babies in Mississippi, which is a really good sign for the species. And that was the first documentation of that population within that river system, or within that creek system. So that was really cool. So you never know what you'll find on these surveys. Here's some good pictures of bear. That was really pretty swamp up in, where was that? I think that's Chambers Creek. And then here's a river red horse. Then we moved on to a less pretty area of Mississippi. We went and dove the entire Big Sunflower River, which is a lot of fun. We found, or of our 82 sites surveyed, we've documented 32 different species, 21,000 live mussels. So there were a lot of mussels in the sunflower, and 8 species of conservation concern were documented during that study. Here's some of our federally listed species. Here's the rabbit's foot mussel again, and these are called sheep nose. So mussels have a lot of really fun names. You should learn all the fun mussel names. Here's the sunflower. This looks like a really pretty kayak, but it was about half a paddle deep that we were kind of just cruising over. Sunflower is very shallow in the spring and summer, or in the summer and fall, so it's really easy to find mussels. You're not diving very deep, and they're kind of just sitting there. But here's some of the big mussel piles we found. And here you can kind of see how deep. This is kind of the middle of the river at this site, and the mussels are just kind of stranded, wandering for deeper waters. But there's a lot of them, so it was a lot of fun. Here's our state geologist, James Starnes, with some washboard mussels. That's a fawn's foot mussel, and that's called a butterfly. So a lot of cool diversity, a lot of cool mussels. There's also Asian carp, if you haven't been there before. And they're not very fun when you're driving a boat. Luckily, we got to kayak for most of it, but on the Lower Big Sunflower, we took our boat out, and we got hit by a lot of carp. So I'll play that again. Some big carp. Luckily, our boat was just fast enough where you didn't get hit 80% of the time if you got up. They kind of outrun the carp as they were jumping, but we did get a couple in the boat. This year we surveyed the big black again, but this was for Piebald madtom. This species is another species that was federally petitioned in the last couple years, so we needed a lot of information for it. Luckily, Matt Wagner, who's now at the Fish and Wildlife Service, did a lot of sampling for the species across the state, but we hadn't seen it in the Big Black River since, where was that? 2007, I think? Yeah, so in the Big Black, we've got several collections from 1973 to 1983, and then our last individual was found in 2007. Since then, MDWFP has gone back and looked for the species on several occurrences, but we haven't found them, and that species was considered extirpated or gone from the Big Black River on the federal SSA because we had this lack of data and because we actually did sample for it and didn't find it. So we decided to go back this year and just see what happened in the Big Black. And of course, the Big Black, a lot of that species range in the Big Black's been heavily impacted by erosion. There's a big truck right there in the bank that fell off from that house. So a lot of it's changed since the 1970s. It's gone through a lot of head cutting events, which is basically just a big erosional wave that moves up the system, and it's completely altered a lot of that habitat. So that species habitat's no longer there, which is why it's kind of disappeared from most of the Big Black. So we went back this year and looked for it. We found 41 different species. We found 5 different madtom species. So the Piebald madtom is a madtom. And we found 22 different piebalds. Actually, we found 28 different piebalds. I don't know why I had 22. But we found 28 piebalds at 6 different sites, and you can see how tiny they are. So we actually went in the spring. All of the sampling for the species in Mississippi has been done in the fall. That's the easiest time to get them. These guys kind of like deeper pockets with flow. Over woody debris, so as you can imagine, it's really hard to pull a seine through an entire tree or kick it. You get a lot of bruises, they're really hard to look for. It's easier when the water's down in the fall. We decided to go in the spring, and instead of detecting adults, we detected recruitment, which is good. They're recruiting in the system, and it's a positive. They are now, known to be within the Big Black River. So that was really fun. You can see how tiny they are. There's 1 centimeter. And there's our Piebald madtom right there. So no adults, but we got juveniles, so good success. And then this year we've also been on the Pascagoula River, which has been one of the most challenging and rewarding surveys we've done. It is a huge system. If you haven't been on the Pascagoula River system, it's a lot of the state of Mississippi. And it's been a blast. So this will be a 2-year survey. But our first year, we did 139 sites in the Pascagoula. We found 9,000 live mussels so far, and 28 species, including 2 species that are federally petitioned for listing. So a good data into the Fish and Wildlife Service. We found the first individuals of the Alabama hickory nut, so another hickory nut species, outside of the Pascagoula River since 2004, and the first individuals documented documented in the Chickasahee River since 1980. And this isn't because people haven't looked or did look for them. This is another area where We just haven't looked for things since 1980, 1970. And then we found 90 individuals of the petition delicate spike. These are the first live individuals in Mississippi since 2004, and the first individuals documented outside of the Pascagoula River since 1970. We found most of our individuals within the Chickasahee River system again, which is where we did most of our sampling. And this is kind of what that habitat looks like for this species. This species is a really cool, I've got one up here, kind of see in the jar. Really cool little mussel. That's as big as they get, so about this big. And they're the delicate spike. So they sit under these boulders in flowy water, and they sit all the way back in. You'll reach your arm about to your shoulder, and you still won't get to them. And you have to keep going back in. Sometimes you have to dive. There was a couple rocks that I was underwater, and up to my hip under the rock. Still couldn't find them. And you have to keep reaching farther, and you'll finally just feel like a McDonald's French fry. The small fry back there, and they're all just poking you, and you have to just get them with your fingertips. It's a really cool mussel. But here's kind of what their habitat looks like underwater. And this is actually really clear for Mississippi. There's the boulder. You gotta reach up and in. And that's the delicate spike. Here's another really clear picture of Mississippi water. This is kind of what a mussel bed, undisturbed mussel bed looks like. You can see their valves sticking up right here. So they're filtering the water, they're cleaning our water. And there's a lot of them. There were thousands of mussels on this mussel bed. So that's really cool to see. Established mussel beds are great for our river systems, and it's a really good sign that you have a healthy river system. Here's some pictures from that survey this year. James Starnes took this photo. Right on the Chickasahee River. And this is, we found a lot of Arctata along here. So you can kind of see these big boulders right here, and these mussels sit up under these big boulders. Here's some of the Arctata we got to find. And there's another boulder system. This is actually in the Leaf River, and this is the first one we found in the survey. I was super excited that we found it. I found it about 10 feet of water under one of these sunken boulders over here. I jumped up and started telling the other biologists about it and got so excited that I dropped it. And spent another 20 minutes looking for it, but I found it again. So here's some really cool formations. And there's a lot of cool fossils you can find along the Chickasahee River. I didn't bring any in today, but this was actually an old marine bed, oyster bed, that had a lot of cool marine fossils and some marine oysters with freshwater mussels on top of those oysters. So that was really cool to see, kind of an ancient mussel bed and then a modern mussel bed. So 2025, we plan to continue this survey. We still have a lot of unknowns about this river system, a lot of areas we haven't sampled, and a lot of information, we still need to gather for this system. So we're kind of looking forward to that. It's going to be a fun survey. And then we're also doing an Alabama shad project in 2025. Same thing, we've got a species, been federally petitioned. We have a lot of questions about this species. It's disappeared from a lot of its range, but it also hasn't been looked for in a while. So we're actually going to go out and do a lot of boat electrofishing this spring and look for this species. So we'll do a range-wide survey of the species in Mississippi, and it'll be a lot of fun to do. And then our 2025 swap, if you're interested in Mississippi's 2025 swap, here's a little QR code, or you can just search Mississippi State Wildlife Action Plan online, and you can kind of get the nitty-gritty, big, thick book of what goes into conservation in Mississippi from MDWFP standpoint. So that's a lot of fun, and it's been fun making it. Again, we have 77 fish listed on it, 45 freshwater mussels, and for the first time we've put some marine species on there, so that's really cool. So some of our random outings we get to do at the museum. Not only are we curating, not only are we going out and doing specific status surveys, but some days we just wake up and decide to do something random and cool. We had found Calvin Rezak. I think he's got a talk in August about this, so I won't go into too much detail, but we were on the Big Black River in 2021 doing our mussel survey, and we found a little species, a little snail shell that we thought to be this species that is extinct now, or was thought to be extinct. So we went back in 2022 and we actually found them. So we rediscovered an extinct species. That was really fun. The species only lives in about 3 miles of river. So it's one of the smallest range species in the entire world. It's the big black rock snail. I've got a little one up here, and there was a magazine article about it in Mississippi Outdoors. But that was a lot of fun, and that was just a random, hey, we know that we think this species might be here. We have a day or two to kill. Let's go out and see what we find. And then here's some articles on that finding. This year we also got to try out some new dive gear. We were bored and decided to go dive the Tennessee River. So we went to Pickwick, which actually is one of the clearer areas of Mississippi, so that's nice. You could actually see some mussels. But we went and tried out all our fun dive gear and just jumped in the lake. Dove Pickwick this summer. There's a big bryozoan Calvin found. There's our 2 technicians with somebody in the water. And this is right outside of J.P. Coleman State Park, so right outside the cabin. So we kind of just got to stay at the cabins and have a little staycation and go dive. A lot of fun, new experience. But this will be important because this training, not only is it, helping us be safe in the water, but it's also adding an extra gear for us to go out and sample some of these deeper areas for these mussels. We can use it now for surveys like hellbender surveys, things like that. So just adding another tool in our arsenal to be able to go out and monitor some of these species. We also get to do species monitoring. We do yearly sampling for a lot of these species, including some populations that we've reintroduced. This species we've reintroduced in coordination with U.S. Fish and Wildlife Service and with Alabama Department of Natural Resources. And we propagate the species over in Alabama every year. We tag them and then we release them every couple years. These individuals actually got released in 2015. You can see their little tag right here. Unfortunately, we found these guys in a muskrat midden, so we didn't find any live ones, but we're really hoping that this population takes off in the next couple years and we'll have a little more success with it. We also do yearly sampling with the Garden Club of Jackson, which is a lot of fun. We go to the Strong River. It's kind of one of our outreach programs, but we aren't just having fun. We're gathering data. We now have a 10-year data set that we can kind of compare. We don't really have a lot of those in Mississippi, but it's very important to have time series data sets like that you can look at an area and kind of see, what's going on, what's changed, if anything major has happened in the last 10 years or so. So it's a really cool, important thing, not only to go out and do outreach, get people excited about fish in Mississippi, but also gather really important data. We've also done a lot of species reintroductions with the U.S. Fish and Wildlife Service, which includes this freckled-belly madtom right here, and then the Yachnapatawpha darter. Can everyone say that? Yachnapatawpha darter? Say it 3 times fast. We collected broodstock with U.S. Fish and Wildlife Service. We reared up this darter at the Private John Allen National Fish Hatchery. Here's the broodstock we collected. Here's a hatchery-raised juvenile with a VIE tag or elastomer tag, and it's a tag basically. It's this little gel tag that's under their skin that glows under a black light. So you can tell that it's a hatchery-raised individual. After a couple years of replacement, U.S. Fish and Wildlife Service replaced a culvert and a creek that historically had these guys, but they had disappeared from. After a couple of years of collecting our tagged individuals, we started collecting individuals that didn't have tags, meaning they were naturally recruiting in the system or naturally spawning in the system, and our job was successful. So that was a really tiny creek, but it was really good to see, and we're hoping to do that with the pearl darter in the next probably couple of decades, because it's a much larger system than what we were releasing Yachnapatawpha darter into. The pearl darter historically was in the Pearl River drainage and the Pascagoula. Unfortunately, it became extirpated from the Pearl, so it is gone from the Pearl River. It is only in the Pascagoula. With U.S. Fish and Wildlife Service, we helped, again, gather some broodstock. We reared them in the hatchery. And last year, for the first time since 1968, pearl darters were back in the Pearl River. So that was a lot of fun. It's going to be a long time before we see if it's a success or not, but we're going to keep that program going. And that's a really cool success, hopefully a really cool success story of this species, maybe coming back, in the next couple decades. Other stuff I get to do at the museum. I get to do some fun surveys like bats, crayfish surveys with Calvin, which is a lot of fun and a lot dirtier than mussel surveys. At least mussels you can wash off. And then we could do a lot of fossil hunting at the museum. So not only do we find fossils randomly on our surveys, but sometimes we'll take George Phillips out onto the Mississippi River or places like that, and we'll look for cool fossils. So that's a lot of fun, extra part of the job. A lot of outreach. Sometimes I'm Santa Claus. He's not that small anymore, but that's my kid. And then we get to go out and meet locals, sometimes, and introduce ourselves the fun way. Some local wildlife too. Local river spirits. Local alligators. Here's my size 11 boot. This was in the Pearl River. We did dive that site, so one of us was just looking out the whole time. And then some really pretty species across the state. So some of our fish species, again, we have 287 species of fish across the state. A lot of them are just, really, really pretty species of fish that you wouldn't think would be in a tiny creek. You pull up a net and all of a sudden there's reds, blues, pinks, yellows, oranges, all sorts of cool colors. This is the blue nose shiner. These are the Gulf darter. This is the Gulf long-eared sunfish, and that's the crystal darter. So these are all really pretty. And all of these are actually in the Pearl River, so you can go out in your backyard maybe and see these guys. Here's some more cool fish species from across the state and across our different surveys. Here's a paddlefish. This is right under the Lakeland Bridge. So, some really cool stuff in your backyard that you just never think about. Some fish pictures by Calvin. Calvin is our resident fish photographer. He takes some really good professional fish photos. See, these are some of the species we've worked with. Here's some good mussel pictures, some juvenile mussels. And some of the insides of some of these mussels are just gorgeous. So some of them are purples, pinks, oranges, and outside too. So mussels are really cool. Different shapes, sizes, colors. I wish I could do a whole talk on just this, but quickly, mussels reproduce by needing a host. So they're parasitic in their larval stage. So a lot of female mussels have a strategy for getting these hosts, and these hosts are usually fish, especially in Mississippi. But they're attracting these fish in to come grab this lure. And you can see right here, the female's gills are full of eggs, or full of larvae called Glochidia. And when a fish like a bass comes in and grabs this lure, she'll actually release her larvae onto the bass. It'll attach and live as a parasite for a couple weeks and hopefully drop off over good habitat. As we have less and less good habitat, that becomes kind of hard to do. But that's their life history strategy, is needing a host. So here is a female mussel luring in. There's a female pocketbook mussel. And it looks like a fish swimming underwater. A little blurry. But it's really cool to see, and you see this in the springtime. A lot of these mussels are luring their hosts in to hopefully deposit their babies. Here is a spawning rough shiner nest. This is really cool to see. Matt Wagner and I went and found this in South Mississippi in 2020, I think. It's been a while, but actual clear water in Mississippi. These guys were spawning over a chub nest, so these guys are called nest associate species. In the springtime, they're out looking to congregate over these chub nests. Chubs build really big rock nests. You might see like a rock pile in the middle of a creek in the spring. That's what that is. And these guys kind of find those nests and just have a mass spawning event over those nests with a couple other species. So it's really cool to see. And here's kind of a slow-mo video of that. And these guys aren't that color year-round. Just in the springtime, the males get really big, pretty, bucked up, and pink. That's really cool. So just some of the cool stuff, again, you can see in your backyard. This was in somebody's kind of drainage ditch. And, with all of this, all of our surveys, all of our studies, collaboration has kind of been the key to success across the state. We have a lot of different collaborators, including U.S. Fish and Wildlife Service, Army Corps of Engineers, State Geological Survey. Other states like Louisiana and Alabama help us out from time to time. So we have a lot of different partners in all the stuff we do. And it wouldn't be possible without that. I can't just go out by myself and do all this. So it's been a lot of fun. We've gotten to work with a lot of different people over the years. So all of our success is due to having a lot of teamwork within the state. And then time for questions. Perfect. Planning them or doing them? All of it? Okay. So with planning them, we start planning surveys, we'll get something in our head, oh, this needs to be done. We'll start that fall or winter, start researching it. Maybe if it's a single species, you'll look at distribution, where it's been, and then you got to propose it. Most of our surveys are funded through grants. It's not the piddly stuff we do just kind of on the state's dime, but our big surveys are funded through grants, and for that you got to prepare a year ahead of time. You have to have justification. You do a little little work towards grant funding, which is a lot of fun. But you write a proposal, you send it off, it gets hopefully accepted, and then you start planning for that survey. So in our downtime, which is right now in the winter, we'll plan our surveys. That spring survey I was talking about, and then the year 2 of the Pascagoula survey I'm planning for right now. Plan it for 2 or 3 months, and then starting whenever the water's warm, so March or April is when we start our surveys. And our mussel surveys typically run March through October. Some of our smaller fish surveys are about a month, but normally a month to 3 months is how long it takes, physical on-the-ground sampling. And then since we do have a repository, we have such a great collection in the back that we are actively adding to, we also have the back work we'll collect a lot of specimens. So we got to catalog and curate all the specimens we collect. That takes about another month or 2. And then by the time winter rolls around, you've got all your data hopefully in that you've entered, your report's done, and then you start planning for the next year. So it's kind of a revolving door of planning and executing. Yes? A lot of practice. So yeah, after about 20,000 mussels or so, you start learning which one's different. And the short answer to both fish and mussel identification is it's because of the way it is. You really do get an eye for it. It's really, for a lot of species in the state, it's easy to go through. Y'all being science students in someone's class have done taxonomic keys before. Really easy to go through a book with a key and key a lot of our general species out, down to the family or the species. But for especially our listed species and especially listed mussels, there's a lot of subtleties that aren't in books that you really have to just handling time and handle something so many times before you're like, oh, okay, this is what it is. And you can just pick it up and be like, oh, that's that. No, because thankfully we have genetics nowadays. So for a lot of our species, we do partner with several different organizations within the state and outside of the state that can genetically identify things for us. So we might have a question for it or for something, and that's kind of how we found the Louisiana pigtoe. The species was on our radar. We didn't know it was in Mississippi, if it was in Mississippi or not. But we kind of just had in the back of our minds, look for something different. We found some individuals that were subtly different from another species in that area. And we're like, okay, we think this is what it is. We're going to get it confirmed. So we got it genetically confirmed. And then, so some of those species you do have to look genetics. Probably the hellbender. That was very surprising. We were just doing our piddly little fish survey and kicked up a mud puppy and it looked different. So if it's federally listed, you got to jump through a bunch of hoops. But for things like the big black rock snail, which is not currently federally listed, so it doesn't have any federal protections, for that species, if we wanted to maybe move it upstream or downstream, because again, it's only in a 3-mile range. So if we want to increase its range in the system, we don't really have to jump through a lot of hoops. We'll kind of write a little plan, kind of what our plan is for reintroducing that species, have a plan for monitoring that species, and then just kind of do it. But for something that's federally listed, takes several years of getting permits, kind of looking at making sure you're not messing with the genetics of the population by introducing something that's maybe not genetically from that area. You got to do a lot of plans for propagation. So you have to have an entire propagation plan that lays out how you're going to hold the species, what you're going to do with that species, how many, if you're going to use any for genetic research, how you're going to cull that species if you need to cull off a population because it's sick or something like that. So you have a lot of hoops you have to jump through if it's federally listed versus just, state concern. Probably drowning. Getting a lot of fun situations. Probably weather is the biggest when we're doing outside research, and here's a thunderstorm we were in. But you get a lot of weather events in Mississippi, especially because we're working in the spring and summer. So hurricane season, tornado season, we're working through that. So you just got to be aware of that. That's probably the biggest hurdle. And then you'll have, 3 weeks of sampling planned, and then all of a sudden we get a hurricane or we get, a big weather event, and the river goes from 3 feet deep to 30 feet deep. So you're like, well, I just got to sit in the lab for the next 3 or 4 weeks. So that's probably, weather's probably the biggest obstacle. So that's a whole 5-hour talk on its own. But in Mississippi, at least, we have a lot of environmental factors. Throughout North America, we have a lot of host issues. So as you add dams, you're removing a lot of host access to those mussel populations. Without that specific host, those mussels die off and disappear. So you have a lot of host problems across the U.S., but I think our main problem in Mississippi is kind of an environmental factor. Most of our river systems, some areas have stable geology, but most of our river systems have very unstable geology throughout the state. And a lot of these areas where these mussels historically existed, even 30 years ago, have been completely altered by what it used to be, whether that be the channel kind of deepening and straightening where you don't have a lot of stable habitat, or banks collapsing in on mussel beds. As you create more instability in these systems, you create a lot less habitat for mussels, a lot less suitable habitat for mussels. So these guys here's our largest species in the state. This is the washboard. Here's our smallest species. And again, and you can see how big these guys get, but it takes a long time to get that big. So this species in particular, I think, lives over 50 years. Same thing with the hickory nut that we looked at. They live over 50 years. So if you think, these guys aren't very mobile. They're not moving around a lot. So they got to sit kind of in the same spot for 50 years. And if that's constantly turning over, they're constantly getting blown downstream, it's very easy for them to die. And then you add in some pollution events, you add in, U.S. Army Corps of Engineers kind of does a lot of or historically did a lot of desnagging where they're moving out, a lot of brush and a lot of trees and stumps and stuff. So that's pulling out a lot of mussel beds. And then just general big rain events, big flooding events like the Easter flood back in the 70s where you have big blowout events, just adds up and adds up to where even if something only turns over about every decade, it's not long enough for a lot of these species to establish. So I think it's mainly a habitat factor. These guys, there's not a lot of stable substrate left. So we're just having, unfortunately, unstable geology has kind of contributed to kind of the state of some of our river systems in Mississippi. So juvenile mussels, just baby mussels. So our juvenile mussels are called, or the larval stage of mussel is called a glochidia, and they're almost microscopic little guys. And then juvenile mussels are kind of, I don't have any juvenile mussels up here, but if this rabbit's foot was about yay big, they're not parasites anymore, it's a mussel, but it's just a lot smaller. So maybe a 2-year-old mussel or so. I think I started out with Steve Irwin, like everybody else my age. And I thought it was really cool that he got to go outside and do some fun stuff and mess around and find cool things. And then got to wear a cool uniform. And I was like, oh man, I want to do that. And then kind of grew up, and I did a lot of coyote research in my undergrad. I liked doing terrestrial stuff, but I liked being outside more. And every time I was out looking for coyotes, I was kind of walking along stream banks and like, I don't know what that is. That's really cool. There's a fish down there. And then just happened to get into aquatics and really liked it and stuck with it and kept going. But it's been a lot of fun. Yes. So different species do contribute to the environment. And it just depends. We've got invasive species that impact it, either positive or negative. Asian clam, for one, is an invasive species. It doesn't really negatively impact the environment, but it's actually kind of positively impacted a couple of our species of turtle, where they're kind of exclusively feeding on that rather than freshwater mussels now because that's what's abundant. So that one's kind of a positive impact. Negative impact would be, of course, the Asian carp in some of our systems. But all of our species as a whole, individually or as a whole, really impact most of our systems. Mussels especially can have a heavy impact on a lot of our systems. Historically, we had massive mussel beds that were intact. You'd have, 200-300 yards of 500, 600,000 mussels. So basically think of it as like a freshwater reef. So they're creating their entire own ecosystem right there. And that's actually increasing habitat stability. Where you might have had a sandy area, you get a mussel bed established. Now it's basically a big rocky shoal, but the rocks are mussels. And they're pulling in nutrients, they're dying there and creating food for other organisms. You've got a lot of habitat for invertebrates and little fish and stuff like that. So again, they are adding basically a freshwater coral reef to the system. So, one little mussel's not making a huge impact, but as you increase it, they're filtering out bad pollutants and stuff like that. They're adding in a massive ecosystem service. And same thing with fish, as you increase your different number of species of fish, you have a lot of different impacts. You create a lot of different food for a lot of organisms and stuff like that. So it's all part of a giant food web. But as you increase diversity, as you increase the amount of species you have, you start getting a lot better, environmental impacts, really positive benefits from that. And as you start taking that away and only have a few species, you start losing a lot of benefits.
One more question.
Sorry, I'm just... Not really a technology. Again, it's just kind of because of the way it is. But the best thing is obviously research and literature. So our books go a long way, and we have an updated book on like Fanny Cook's 1959. This is great for what it was. But these all have big taxonomic keys. It lays out this, this shape gets you to this number of species. Then you look closer at this shape, then you get closer to this shape, and you'll get down to 2 species, and it'll say this one has this one has this. So that's the easiest way. It's basically just a big key that you can follow all the way down until you identify what you have in your hand. So that's the easiest way with any species, is find a taxonomic key that can help you out. Yeah. And then...